All right. So we are here. All right. So after Leeuwenhoek, then there were two other guys that later added to our knowledge of the cell theory. Okay. First, we have Francesco Redi. Francesco Redi. Okay. Now, what he did, he proved that living things cannot be produced from non-living matter. Okay. I think this guy over here is Redi. And then over here, this is Louis Pasteur. Okay. But what Redi did was this. At the time, they believed that sometimes living things kind of, sort of, poofed into existence. And here's why. Remember how we've been talking about with the Age of Exploration, how in Europe they didn't have a lot of spices, they didn't have a lot of salt, and they had no way to preserve food and keep it from rotting, right? There's no refrigerators, there's no freezers, things like that. So any food they had that they left on the counter, what would happen to it? Well, over time it would rot, right? Rotting food, if it rots long enough, will eventually produce maggots, okay? Little baby worms, basically, that eventually turn into flies. The maggot's job is to eat the decomposing, rotting food and help it break down, okay? So all they noticed was, hey, this is a piece of meat sitting on the counter, and now there's living stuff crawling around in it. So to them, it looks like this piece of meat is, poof, creating living things. Okay, so Francesco Redi, however, said there's got to be some explanation. You can't just have something non-living randomly create living things. Go ahead. So what he did, he designed an experiment. He took a couple pieces of raw meat, and he had a bunch of jars. He put a piece of meat in each jar, okay? He covered half the jars with a lid and left half the jars uncovered, okay? After a few days, what he discovered and what he noticed was the jars that had the lid screwed on top, that meat did not produce maggots, but the jars that were uncovered did produce maggots. Can anyone explain why? Jacob, what do you think?
I think the reason is that the ones that didn't have cover were exposed to air.
Okay.
And so that was able to make it rot.
But, so the air produced the living things?
No, I'm just saying it helped.
Okay, well, there was a difference, obviously. The ones that didn't have the cover on it did produce the maggots. Okay. What got into the jar that created the maggots? Not the air. It can't come from non-living things. Riley?
Germs.
Not the germs. Connor?
So flies. What they would do is they would land in the jar, lay a bunch of eggs in there, and then over a few days they will hatch, become maggots, and over another few days they will become flies.
Very good. Okay. The reason why the uncovered jars had maggots wasn't because that meat just poof and produced maggots. It was because the flies were able to get into the meat. They landed on, right? Flies buzz around rotting food, right? Mm-hmm. Yes. They landed on the meat. They laid their eggs, and that's where the maggots came from. Okay? Proving that the maggots can't just come from non-living things. They had to come from something that was already living, which was the flies. Okay? Hence why they got into the uncovered jars and not the covered ones. The flies couldn't get in. Yes, Matthew?
Like once, at the end of Louisville Lake, Louisville Lake Park, so I saw this fish next to the shore, and I was attempting to hit it, knock it into the water, and I flipped it over, and on the bottom side there were hundreds and hundreds of disgusting maggots.
Ew.
And it started crawling everywhere.
Ew. Yeah.
Some were like a millimeter, some were like 2 inches.
Ew. Ugh. Yeah. Okay, so Francesco Redi, he proved that living things have to come from something else living, can't just poof into existence. And then we have Louis Pasteur. He kind of added on to that by saying that cells can't just come from any living thing, but cells only come from other living cells. Okay, they can't just poof out of anything living, but the one cell splits and reproduces to create more cells. Okay? Every living thing once started as one cell. Okay? That one cell then further divided and split and multiplied to create the trillions and trillions that make up the organism. Okay? Fun fact, raise your hand if you drink milk. Okay? Louis Pasteur, you drink pasteurized milk, meaning it's been treated chemically and a lot of the antibiotics and stuff have been taken out. He's the one that created that. Pasteur, pasteurized milk. Has nothing to do with cells, but it is interesting. Okay? Luke?
So it's kind of about the meat and how the blood goes.
Okay.
So it's a little bit gross, but you know how whenever dogs can find the flies and everything on there?
Mhm.
You know how flies are all over it?
Mhm.
Like, are they laying eggs?
Mhm. Usually. Or they're trying to get the nutrients from it. Yeah. Help break it down. Yeah, flies are gross. All right. So now we're going to fill in the 3 parts of the cell theory. Let's see if we can do it before I give you the answers. Number 1, all living things are made of, Aiden? Cells. All living things are made of cells. Number 2, cells carry out the functions needed to support what? Jacob? Life. Cells carry out the functions needed to support life. And then number 3, cells come only from, Layla?
Other cells?
Other living cells. Cells come only from other living cells. So here's the answers. Number 1, all living things are made of cells. Number 2, cells carry out the functions needed to support life. Number 3, cells come only from other living cells. Matthew?
Do some cells eat other cells?
They can.
Cannibals.
Or destroy other cells, yeah.
Cannibals.
Connor?
Yeah, with Matthew's question, it's kind of like how the white blood cells go around eating the germ cells.
Correct. Aiden?
So, technically, they're not being cannibals, but they're eating a different type of cell.
I suppose so. Chloe?
So, but it's the same species. But okay, but if the white blood cell eats a germ, would the white blood cell get sick?
No, because the materials inside the white blood cell kill off the sick part of the germ cell.
So there's a germ cell that's a healthy germ cell left.
There can be, yeah.
And then so do red blood cells, do white blood cells ever accidentally eat red blood cells?
I don't think so.
The germ cells?
I don't think so. I doubt it.
No,
All right. We'll come back to this video, the history of the cell theory, in a little bit. But we're going to go ahead and move on to the 2 different types of cells. Okay? So now that we know a little bit about the cell theory, now we're going to talk about the 2 different types of cells. Okay? Someone raise your hand and tell me, what are the 2 types of cells? We have discussed them already in the classification unit. Let me give you a hint. Brian?
Eukaryote, prokaryote?
Very good. Brian, which of these is eukaryote? Left or right?
Left.
Left. Okay? This is the eukaryote cell. This is the prokaryote cell. Why is this one the eukaryote cell? Manny?
Because they have like a nucleus.
Very good, because the DNA is wrapped up in the nucleus. Okay? The prokaryote cell does not have the nucleus, but instead has all that DNA kind of just hanging out in the middle there. Okay? So, for your definition in your notes, you're going to write this whole first bullet point here for the definition of prokaryote cell. Remember, the way you remember it is pro rhymes with no. A prokaryote cell has no nucleus. So we're going to write this. An organism with no membrane-bound nucleus and no membrane-bound organelles. That is your definition for prokaryote cell. An organism with no membrane-bound nucleus and no membrane-bound organelles. Once you've written that, you're going to draw a model of a prokaryote cell. which will look something like this. You have your cell, and then we add DNA, but it's all kind of just free floating through the cytoplasm, through the fluid inside of it. It's not wrapped up into a nice, neat container. Looks like spaghetti, we said. Okay. Once you finish that, we're going to write the definition for eukaryote cell. A eukaryote cell is an organism that has a nucleus and has membrane-bound organelles. And then we're also going to draw a model of that one. This one will look a little different though. So we're going to have our cell. Okay. Then we have the nucleus, the defined membrane-bound nucleus in the middle with all the stringy DNA. But nice, neatly packed into the nucleus. Okay. Now, because it's eukaryote, it does have a membrane-bound nucleus, and it also has membrane-bound organelles. So just like our body has different organs throughout our whole body, right? We have a section where our stomach is, we have our liver, we have our heart, we have our lungs. And a eukaryotic cell has organelles that are like tiny little organs, okay? So they have ribosomes, they have the endoplasmic reticulum, which we're going to learn about. They have Golgi bodies. They have mitochondria. They're shaped like little peanuts. Okay? So it has lots more complex structures throughout the cell in a eukaryotic cell. Prokaryotic cell does not have that. Okay? Much less developed, much smaller, and much less complex. Layla? No, but just at least make sure you have the nucleus. Luke? No. We're going to go on to the next page in a minute. Matthew?
Is this actually a dual-celled organism?
Duo-celled? Dual-celled? I don't believe so, no. It's either one or many.
Would many count as two?
No. Aiden?
So...
Okay, go on to the next set of your notes. You should see a page that has two columns, the prokaryote characteristics and the eukaryote characteristics. Okay, I'm going to flip this over to the dot cam so we can fill those out. Yes?
Actually, the cookie can superglue one cell to another cell.
Somehow I don't think that would work. You'd probably kill both of them. No summary for this one today.
Yes.
All right, but we are going to look at the next page, okay? So you should see two columns, one for the prokaryote characteristics, the other column for the eukaryote characteristics. Okay. So, first we're going to talk about, now this part's kind of tricky because this next little bullet point that we're going to write talks about the prokaryote cells were the first to evolve and the eukaryotes evolved later. Okay, now that is specifically for scientists that believe in evolution. Personally, I don't, but there are some that do. Okay, but what they're getting at is these cells are much less complex. So these probably came first, if you believe in evolution, and the eukaryote, or the more complex, developed from that. Okay, so prokaryotes, first cells to evolve. Eukaryotes evolved from prokaryotes. Next, the prokaryotes have no nucleus, while the eukaryotes have a nucleus. Okay, obviously you can see in the picture, the prokaryote, all these little stringy lines, that's the DNA, but it's not wrapped up in a nucleus. So no nucleus. Over here, here's the DNA, all stringy in there, And it is surrounded by a nucleus. It is contained in one little circle. Riley?
Never mind.
Okay. Next, hereditary information. Hereditary information is a fancy way of saying the DNA. So, for a prokaryote cell, the DNA is contained within the cytoplasm. Okay? Cytoplasm is like the liquid kind of jelly stuff that fills the whole rest of the cell. You kind of have to think of a cell like a bubble. Okay, like a big jelly-filled bubble. All right? Everything inside of it's kind of floating around in that. So, since prokaryotes have nowhere else to keep the DNA, it's just floating in the cytoplasm. But in eukaryotes, the hereditary information, or the DNA, is contained where? Jacob? Good. Within the nucleus. Yes. Chloe?
About animal cells and stuff earlier in the year.
Mm-hmm.
Well, do you imagine the cells of the cell city?
Yes, we're going to get to that. Okay? Yes.
So, do humans only have like one of the type, or do we have prokaryote and
Great question. Every single one of our cells is eukaryotic.
so
No.
prokaryotes?
I don't believe so. I mean, unless you have like germs or bacteria, which you do, but every part that makes up you as a human is made from a eukaryote cell. Okay? Yes?
What if a person had a prokaryote?
Then they wouldn't be classified. They wouldn't be possible because they're not. To be in domain eukaryote where we are, you have to have a eukaryote cell. Okay? So, speaking of that, examples. For prokaryote, you can find prokaryotic cells in the organisms within domain archaea and bacteria. Well, these are domains. Archaea bacteria would be the kingdom. For archaea, yes. And then eukaryotes, you could find examples in kingdoms, Animalia, Protista, Fungi, and Plantae. Yes.
This is where we had to learn about the animal kingdom first.
Partially, yes. All right. I'm going to show you a video in just a minute that compares the two types of cells. But first, turn to the last page, the Venn diagram. You should see something that looks like this. It should say prokaryotic versus eukaryotic cells. It should be right after that, the ones we just did, the two columns. Okay? All right. Turn to the back of this paper. It should look like this. All right? There's a couple things we're going to write first. The top little half semicircle, okay? I want you to label that pro for prokaryote. This little half circle, that's where you're going to write the characteristics of prokaryotes. Okay, I'll explain in just a minute. At the bottom half circle, I want you to label euc, E-U-K period. That's for eukaryote. All the characteristics for eukaryotes you'll write in this half circle. Now, if you write something in this box and think, hold on, wait, that's not true about eukaryote. That's true about prokaryote too. That is a characteristic of both. That goes in the middle section. The part where the two circles overlap that I highlighted, okay? That's where you're going to write characteristics that are true of both. Okay? Now, couple directions. At the top, I want everyone to write these 3 stars. Number 1, this does not need to be in cursive. It's a small little space. I'm not too worried about cursive. If you want to write it in cursive, you're more than welcome, but you don't have to. Number 2, this also does not need to be in complete sentences. Again, small space. Not too worried about it. If you want to write in complete sentences, you totally can. And then the last direction, you have to have 4 or more facts in each section. That's what 4 plus means. So you have to have at least 4, but you could have more than that if you wanted. Now, I am going to caution you. Do not give me a stupid answer. What I mean by that is this. Do not write in the middle, these are both cells. That's not going to count.
I was gonna remember that.
Okay?
They're tiny.
Huh?
But the cells are tiny.
Microscopic. You could say that. Now, I'm going to show you a video here in just a second that goes through quite a few more characteristics than what we have in the notes. So I would listen very carefully because you're probably going to want to use that in your homework. Okay? First, I'm going to show you the cell theory video, and then we'll do the prokaryotic video. But are there any questions on how to do this homework? Brooklyn?
Whenever you show us the video, can we write stuff down that we can just...
Sure. Sure. All right. Okay. Hold on. Let me stop this recording real quick. Yeah.